Good afternoon and welcome to the UPMC Children's Hospital of Pittsburgh Center for Rare Disease Therapy webinar series. I'm Jerry Vockley. I'm the director of the Center for Rare Disease Therapy here at Children's Hospital and the chief of medical genetics. I'm going to be talking to you today about fatty acid oxidation disorders and specifically novel therapies for inborn errors of fatty acid oxidation, utilizing a personalized medicine approach to help us provide better therapies. Just to start off with, I show you my research funding and conflicts of interest, some of the companies that I consult for. To understand what we're going to be talking about today, I'm going to drop back to some very basic biology, and that is what is called the central dogma. This is the transfer of information from DNA, the storage material of information in our cells, to proteins, the part of the chemicals in the cell that carry out the functions. So our information is carried for the most part in our nucleus, in every cell, on DNA. That DNA has to first be made into a copy called RNA, and the RNA carries the information then to the rest of the cell called the cytoplasm, where it's made into proteins, a process called translation. And if you have a mistake in your DNA that either leads to it not being transcribed properly, read properly, makes the RNA unstable, or makes a protein that is unstable or doesn't function properly, you have a genetic disease. We're going to be talking today, of course, about energy metabolism, and I'm going to start off by showing you some very key interactions that are important to recognize when we're talking about fatty acid oxidation disorders. The process of fatty acid oxidation involves the release of stored fat from our tissues into the bloodstream. Those fats are brought into the cell. They're transported into a section of the cell called a mitochondria, and then they are burned through fatty acid oxidation. Fatty acid oxidation is a four-step process that is specific for the size of the fat that's being burned. The fats that are stored in our body are long strings of carbon atoms. Usually they're 16 to 18 carbon atoms long. And the fatty acid oxidation knocks 2 carbons off at a time and generates a compound called acetyl-CoA. For long chain fats that are from about C16 all the way down to about C10 to C12, we call that long chain fatty acid oxidation, and they're inside the mitochondria. The reactions are carried out largely by very long chain acyl-CoA dehydrogenase, the LCHAD, and the mitochondrial trifunctional protein, which includes LCHAD, long chain 3-hydroxyacyl-CoA dehydrogenase. Now, when that acetyl-CoA is released from fatty acid oxidation, it can do one of two things. It can go to make ketones. That's the fuel that the body can use instead of glucose if you're fasting, or it can go into the TCA cycle. The TCA cycle, also called the Krebs cycle, TCA stands for tricarboxylic acid, makes additional fuel that, in addition to the fuel that comes from fatty acid oxidation, goes into a third pathway called electron transport chain. And the electron transport chain is actually now the set of chemical reactions that make ATP, the fuel that most of the biochemical reactions in the body, in the cell, need. The fact that all three of these interact means that they can also overlap to some extent in their clinical symptoms, and it may even turn out in some situations that the problems related to, for example, generation of ATP from fatty acid oxidation are actually the ones that are the most pertinent to the clinical condition. Let's take a further look at those mitochondria that I mentioned in the last slide. Mitochondria are these very specialized structures that occur inside every cell, and there are a handful to hundreds of these in every cell. And they have this bean shape in cartoon form, but in reality, they're very much more complicated. But we're going to look at this to understand the basic principles. There's this outer covering called the outer membrane, and this inner covering called the inner membrane. And the inner membrane has this very unique structure, here cartooned as these pouches of the inner mitochondrial membrane. You can actually even see them here in an electron microscope. And most of the respiratory chain, the electron transport chain that I mentioned previously, and fatty acid oxidation for long chain fats is right down here in this area called the cristae. And one thing that I'll mention now, and we'll come back to it closer to the end of the talk, is that to be able to make these very dramatic bends, there's a special fat, a special lipid that the inner mitochondrial membrane needs called cardiolipin. And just hold on to that for a little bit and we'll come back to it. All right, we already talked about that. So one of the things that I've worked a lot in my laboratory where I research fatty acid oxidations has been to characterize how fatty acid oxidation interacts with the respiratory chain. And this is a cartoon that shows that. The respiratory chain is composed of 4 large protein complexes called, simply enough, complex 1, 2, 3, and 4. They interact together in this inner mitochondrial membrane right here. And this helps make the cytochemical reactions very efficient because everything's all together. And over the last 10 to 12 years, work that I've done has shown that the fatty acid oxidation proteins also interact with this electron transport chain here at the inner mitochondrial membrane. And so this is one large complex that is necessary for efficient metabolism. What does that mean to clinical symptoms? Well, think about a house of cards. If you have a house of cards and you pull this top card off, well, you're not going to do very much besides lose that top card. However, if you pull off something down here at the bottom, what's going to happen? Well, the whole thing is going to fall down. And so you can think about that complex of proteins interacting in a similar way. You can have mistakes that just cause one of those proteins not to function properly, or you can have mistakes that disrupt the whole complex and make it fall apart, and that keeps it from not just the fatty acid oxidation working properly, but those other cycles get involved as well. I mentioned that most of the fats that we store in our body are strings of carbons, and those carbons are those carbon chains are 16 to 18 carbons long. Here's a picture of a 16 carbon chain called palmitate or palmitic acid. Look on your bottles of shampoo. You'll see this as an ingredient, but it's also a storage form of fat. When we burn 1 molecule of palmitate, you get 123 molecules of ATP. It's a very high energy set of reactions. In contrast, you only get about a quarter of that if you burn a molecule of glucose. So this is a way of storing and harvesting high energy when you need it. Now, if this doesn't work, if you break it right at the very beginning at the long chain fatty acid oxidation, not only don't you make it, but you actually spend a little ATP to get the whole process going. And you can see that this is a major energy deficit in your body. And to try to compensate for that, the body revs up the other cycles that it has available to it. In the mitochondria, that's the TCA cycle. And so when the TCA cycle is going at high speed, because the fuel that the electron transport chain needs to make ATP is not getting to it. The TCA cycle goes and it goes and it goes and it goes. The fuel is acetyl-CoA, which we can still get from C8, medium chain triglyceride. and that can come into the mitochondria very readily. It bypasses these enzymes, and so you can make the TCA cycle work with that. The problem is that even with the TCA cycle running off of acetyl-CoA, eventually the system seems to stop. And why is that? Well, Dr. Charles Rowe, a number of years ago, was the first to suggest that what's happening is that acetyl-CoA is only one of the things that this cycle needs to work properly. This is C2. It also needs odd chain carbons, C3, and that is shown right here at this stage of the cycle. And so if you're just giving C8, you run out of C3 as this keeps running and running and running. So Dr. Rowe thought, well, let's give C7 instead, where you can make 2 molecules of C2, 1, 2, and have a C3 left over. And that's triheptanoin. And that whole process is called anaplerosis, and using it for therapy is called anaplerotic therapy. This now has been used for close to 20 years, and I'm going to show you some of the information that's now allowed us to bring this to the FDA for approval in the coming year. First of all, Dr. Melanie Gillingham and I at the University of Oregon Health Science Center did a clinical trial where we looked at where we compared patients who were taking triheptanoin or were put on triheptanoin compared to patients who were put on C8, medium chain triglyceride oil, in a blinded fashion. And we did a very complicated set of clinical tests. Patients came to the hospital, did some things at home. Not going to spend a whole lot of time to talk about that. And then 4 months later, we brought them back in the hospital and repeated that. And what we were able to show was that they showed that they had improved cardiac function, even though all of the patients in this study had normal cardiac function to start with. The left ventricle, the part of the heart that pumps the blood out to the body, was improved significally with triheptanoin compared to C8. And Dr. Gillingham had even shown that C8 improved over a high carbohydrate load. So the improvement of C7 over carbohydrate is even more significant. Well, that's a duplicate. We didn't see any clinical changes in those individuals because they were well to start with, and we were only following them for 4 months. And so we really couldn't make any comments about that. But at about the same time, I started working with a company, Ultragenics, who got interested in triheptanoin and agreed to take it on and develop it further as a therapy that they would ultimately be able to market commercially. And so our next study was a study of 27 patients who had previously been treated with MCT oil and then were switched over to triheptanoin. We didn't keep anybody on the MCT oil for this study. And you can see of the 27 patients, almost everybody had a history of rhabdomyolysis, those painful episodes where you have muscle breakdown, and significant muscle pain and exercise intolerance. Most of them had a history of hypoglycemia, and about half of them had some problems with their heart. We've looked at the data. We've now had patients treated in this study for 78 weeks. And if you look at the overall rate of having a major clinical event defined as an episode of rhabdomyolysis, hypoglycemia, or start of or worsening of cardiomyopathy, that overall patients on triheptanoin had a clinically, statistically significant improvement in their clinical event rate compared to when the 12 months prior to their starting that therapy. Hypoglycemia was essentially eliminated. Cardiac events were cut by a little over 2/3, And rhabdo was the least affected by this treatment. Patients were better. They had less episodes. By about half, those episodes were less severe and they were shorter, but they were still happening. So these data are the data that we're now taking to the FDA with Ultragenyx, hopefully in this coming year, to ask for approval for general use of that compound. But that still leaves us with a question: if patients on TriHep are still having rhabdo, why might that be? Well, one of the things, I'm sorry, let me just quickly tell you that the safety of this drug was quite good. We saw a fair amount of GI symptoms, mostly because we were increasing the dose a little bit compared with most patients on MCT oil, but basically nothing that we didn't see on patients who had MCT oil. And so we think this is going to be a very safe drug. But if it's not completely fixing the rhabdo, the question is, what's going on? Why are patients still having muscle symptoms? Because with the TriHep, we think we're fixing the energy deficit. Why are we still having problems? Well, one of the other things that we know about that can cause rhabdo in patients is inflammatory muscle disease, inflammation of the muscles that cause it to break down, unrelated to energy deficit. So I did a quick study. I was able to go into my freezer and pull out 16 samples from patients that I'd seen over the years who were both on either on MCT oil or triheptanoin, and compare them. And it turns out that it didn't matter whether they were on MCT or triheptanoin. They still had evidence of inflammation in their blood and in their white blood cells. In this picture, what you can see are 7 different what are called cytokines, inflammatory molecules that we can measure in the blood, and green is normal. Anything other than green is abnormal, and anything that's red is really abnormal. And so what you can see is that everybody in this study had almost everybody. Here's one who just barely had a little bit of abnormality, but almost everybody had some evidence of inflammation. And so you can think of this as somebody sort of standing on a cliff looking over at the river on the bottom. You're not having trouble yet, but all it takes is a little shove, and you're falling over the cliff. Well, what can we do about that? Why is that happening? Well, we think it's related to that original picture that I showed you of those 3 cycles not working properly, and that house of cards. So if you have a change in your fatty acid oxidation proteins that affect the respiratory chain, the electron transport chain, what happens is that electron transport chain, we think, is now less tightly packed. And when that happens, the oxygen that it's burning for fuel to make the ATP the intermediate compounds called reactive oxygen can leak out into the mitochondria and cause damage. That's called reactive oxygen or superoxides. And here, one of our patient's cells, these are skin cells that we grow in the laboratory, taken from patients when we first diagnosed them with fatty acid oxidation disorder. And here, what you can see is these damaging oxygen species, superoxides, in this patient's cells are very much higher than in a control cell line. And this has been true for almost every cell line that we've looked at from patients with long-chain fatty acid oxidation disorders. So we think we're seeing something here that may help explain the rhabdo episodes and will allow us to treat it with a different drug that can potentially help to control this. This is an antioxidant, something that controls these reactive oxygen species. And you can see that here in the control cell lines, the normal cell lines, doesn't really do anything. But in that cell line I showed you before, from a patient with VLCAD deficiency, it drops the reactive oxygen species. It doesn't normalize it, but it drops it by a lot. And we think this will translate into clinical improvement in patients who are having a lot of rhabdo. We're not at the point where we're ready to put this into patients yet, hopefully another year or two, but we're moving forward on that. Just to show you that this is not something that is specific to VLCAD. Here's a patient, cell line from a patient with LCHAD deficiency, and you can see these reactive oxygen species are also elevated. Now, I didn't include the picture here that shows response, but it also does. These cells also respond to the reactive oxygen, to that antioxidant. So we should be able to take cell lines from patients, test them, and if the reactive oxygen species is up, then use one of these antioxidants to help things. We're also looking at a way to help stabilize that house of cards. So if your mutation, if the change in your gene causes that house of cards to fall apart, what if we could give you something that helps stabilize it? And so molecules that help proteins fold and hang together are called chaperonins, and we're developing a series of those that we're now testing in cells to see if we can improve the fatty acid oxidation in patients. So here are cell lines, again, from a patient with VLCAD deficiency, this one here, and one with trifunctional protein deficiency, this one here, treated with a drug called trimetazidine, TMZ, to keep me from having to trip over it too many times. And that, when we treat cells with an increasing amount of it, allows both a TFP and a TFP deficient cell line and VLCAD and a VLCAD deficient cell line to become more stable and hang around longer and increase activity. So this will be another type of therapy that we could potentially use, and this one will only work if the original change in these cell lines is causing that house of cards to fall apart. As we build more and more tools to help treat fatty acid oxidation disorders, we're talking now about not just treating everybody but with the same thing, but personalizing it to your specific version of change that might be in your causing your disease. The next approach that we're using is something called a transcriptional activator. So if you go back to that very first slide that I showed you, remember I said you got to make a copy of your DNA into RNA, which then gets made into protein. Well, transcriptional activators cause more activity and allow the cell to make more RNA from the DNA. It makes that process, revs that process up. And so if you have a change in your protein that leads to a stable protein and a little tiny bit of activity, if we can rev up your gene and get more of it made, more of that protein made, a little bit more of something that's a little bit active is going to improve things, right? So here you can see the drugs that have a fancy name. They're called PPAR delta agonists, and this graph shows a measure of the mRNA being made from a cell line treated with a transcriptional activator, and you can see that starting down here and then increasing the concentration, you get more RNA, more VLCAD protein, more VLCAD activity, and we can measure how well these cells utilize that fat I was talking about before, palmitate, and these cells can use it better. So if you have a protein that's stable, has a little bit of activity, we can treat you with this drug and potentially improve your metabolism. We're just getting ready now to start a clinical trial hopefully in April, just a couple of months away now, with a company called Reneo, who has developed one of these PPAR delta agonists, and is working with a number of our fatty acid oxidation consortium sites around the country, and you'll be able to get more information about that on clinicaltrials.gov, and we'll also have it on our consortium network website that I'll get to in a second. Now, testing your memory here, remember I mentioned cardiolipin, this special fat that lets the inner mitochondrial membrane bend. If this membrane doesn't bend, the cardiolipin that sticks to the respiratory chain proteins and allows them to stay in the membrane goes flat, the mitochondria become unstable, and the cells die. Cells dying is what causes rhabdomyolysis. So here's another possibility for cells, for energy in cells not working properly. Why is this important for fatty acid oxidation? Well, the structure of this fat, this cardiolipin, is such that it's affected by reactive oxygen. Instead of having the correct shape, it now takes on a shape that won't let the mitochondrial membrane bend, and so a fatty acid oxidation change that causes increased reactive oxygen is going to affect cardiolipin. This whole house of cards thing again. It's all falling apart. Well, can we fix that? Well, we have a drug that it interacts with cardiolipin that has been affected by reactive oxygen and forces it back down into the correct shape. And here you can see that LCHAD cell line that I showed you before, with this higher level of reactive oxygen compared to control cells. Now we treat it with this cardiolipin drug, and it completely normalizes it. And so if you don't respond to one of those other drugs, or you have, it may well be that this is a drug that's particularly useful for LCHAD, because LCHAD, as it turns out, is an enzyme. It doesn't just do fatty acid oxidation, it actually helps make cardiolipin. So maybe this will be a good drug for LCHAD. Again, we're doing a lot more laboratory testing now to look at all different kinds of fatty acid oxidation cells, and add another tool to our bag of tricks to be able to treat individuals with fatty acid oxidation disorders for their particular flavor of the disease, not just everybody the same way. All right, if we can make more mRNA from a drug, that's great, but there's technology available to actually give you more mRNA. And what you can see here is an experiment where we take cells with no VLCAD activity, no mRNA, and give it mRNA from a company called Moderna, and we get gangbusters activity. When we do this in a mouse model of VLCAD deficiency, we can get lots of enzyme. When we do this for a mouse model of MCAD, we get lots of activity. But it just gets into the liver. So this isn't going to help the heart. This isn't going to help the muscle, and that's part of the problem with this therapy, and why it's a little bit further away from clinical use. We still have to figure out how better to target it to the right tissues. I should say that gene therapy does the same thing. We can get it into cells, we can get it to express, we can give it to animals, we can get it into liver, but it's hard to get it elsewhere. So gene therapy kind of falls into the same category, but it's currently in the works. Now, finally, for those of you who have MCAD deficiency, I don't want you to feel like nothing's going on. We are working on a new therapy for MCAD deficiency. It turns out that about 90% of the mutant genes out there for MCAD deficiency have a single mutant change, one change that is common to everybody. If you work out the genetics math, it turns out that 3/4 of I'm sorry, 3/4 of the mutant mutations are this one mutation, and the 90% that I was saying before is that 90% of individuals who have MCAD deficiency have at least one copy of this common mutation. But we have a drug that when we give it to cells with MCAD deficiency, increases the MCAD activity because it's able to bind to the protein and act as one of those chaperonins. It helps the enzyme fold properly. And it turns out that this enzyme, if we can fold properly and make it stable, actually works quite well. So this is a drug that we've tried in very early clinical trials. We're getting ready to hopefully get another clinical trial, a phase 2 clinical trial for this one later this year. So watch the clinicaltrials.gov or our Inform Network website, and we'll let you know when this clinical trial is up and running. I have a lot of people who help me out with these studies. Folks that you may recognize here, Dr. Gillingham, others here at the University of Pittsburgh, who are critical to these studies, and I thank them. And before I move to questions, I just want to remind you of, well, there's a thank you, and I think, did they put on, yep. This is our lovely Children's Hospital here in Pittsburgh, and you'll be able to go and ask questions on your webinar tool. Go to webinar tool, but I want to remind you of two resources for you. First, our CRDT, our Center for Rare Disease Therapy. You're always welcome to contact one of the staff here, and we can answer questions as best we can. But we have now an international collaboration of centers around the world who treat fatty acid oxidation disorders, called INFORM, the International Network for Fatty Acid Oxidation Research and Management. That, if you write it out, you can get INFORM from the first letters. And the network, the website is called informnetwork.org, and there's a family page there. It has a lot of information that you can use to hopefully educate yourselves. But there's also an Ask the Expert page there, or function there, that'll get your questions to one of the INFORM physicians or scientists who can answer your questions. So here's the contact information for the Center for Rare Disease Therapy, rarecare at chp.edu. We'll get you the staff here, or the phone number. And then I am going to take some questions. Now, unfortunately, you can't see these, so I have to read them, and it's very small print. So I'm going to look a little silly coming close to the camera here and read these to you. So the first question, don't have a name here. It was just oh, it's from Sarah. There we go. Oh, that just says this is a test. It worked, Sarah. It's one of our staff here. I'm sorry. The first question was sent in via email to our CRDT staff. I'll get used to this eventually. Are there any studies being considered investigating the possibility of using a combination of high-dose vitamins to treat mitochondrial dysfunction and oxidative damage? And I'm just going to abbreviate that. And we had another question that was very similar to that. That came in over the network. And you've seen my information on reactive oxygen being an issue in fatty acid oxidation disorders. The jury is still out on all of those things you hear about treatment for mitochondrial respiratory chain deficiencies. There's a whole long list of drugs or supplements, a couple of dozen of them that people use in various combinations. And most of those, when we test them on cells, don't work in our system. they just don't seem to be, they don't get to the mitochondria well enough to be able to make those work. Now, I know that there are some people who have tried them, and I've talked to them, and they think they've helped them. I know some physicians who have tried them because they didn't have anything else to do for a patient who was particularly difficult. So this is just one of those where we hope our informed network will ultimately help us try these things in a fashion that lets us answer those questions scientifically, and we'll be able to give you better information. Let's see, the next question says, I understand that LCHAD is an inherited disease, but I also assume that at some point and for some reason one of my ancestors acquired this condition. The question is, what are the causes for this mutation to happen? And that was sent in from Alejandro. Well, Alejandro, there are two ways to get for one of your cells to get a mutation. You either inherited it from a parent, as you said, from a family member, or it happened in the egg or the sperm cell that came together to form you for the very first time. And that's called a new mutation. And when we look at genetic studies across the board, we know that people carry changes in their DNA that aren't present in either of their parents, hundreds of changes across the genome. And so this happens all the time. The mechanism, well, it could be anything. Any of these reactive oxygen things that we talked about, the DNA that goes to make an egg or a sperm cell has to be copied from other DNA. And if you think about the monks back in the Dark Ages copying manuscripts, every time you copy something, you can make a mistake. And the cell does that. It has very sophisticated ways of trying to correct those mistakes, but occasionally, about 1 in a million times, it doesn't get corrected properly, and so you end up with a change. So somewhere along the line, that change either happened in one of your relatives, or it happened in one of your parents, and that's as close as we can come. Let's see. From Stephanie. Hi, Stephanie. I'm trying to understand the concept behind why using MCT oil can lead to increased demand on the TCA cycle. About 2/3 of the energy that comes from burning a molecule of fat comes from making that acetyl-CoA that goes into the TCA cycle, and about 1/3 comes directly from the reactions of fatty acid oxidation. And so if you cut out 1/3 of your ability right from the start to be able to make anything, and now you give C8 and C16, so you've cut your capacity by half again, the TCA cycle is going at full speed, and you just can't keep up with the demand. You can get lots of acetyl-CoA from MCT, but eventually you run out of that propionyl-CoA, that C3 that's critical to keep the function running. And if you can't make energy from the fatty acid oxidation, you can't make energy from the TCA cycle, you can't make energy, and that's where you run into trouble. Let's see. John says, my son rather is 3 years old with MCAD. As he ages, are there foods to stay away from? For example, foods that are high in fat and carbohydrates. This is a bit of a complicated question, and I'll try to keep the answer simple. For you MCAD folks out there, the answer is probably not. If you remember, what happens when you eat is you don't actually burn that fat right away. You store it. The body signals, I'm eating, I'm going to store energy. And it's only when you stop eating, or you get sick, or you're running, or something that requires a lot of energy, that you now call on your stores to burn fat. And so your intake doesn't affect you very much. Now, that said, most Americans' diets are too high in fat, and I recommend to all my patients a heart-healthy diet, 30 to 35% of calories from fat. And an MCT and MCAD deficiency, you can't use MCT oil. That's toxic to you. That's C8. That's exactly what you can't burn. So you got to use long-chain fats, but you can only use them about half the way. You get down to C8 and it's and you can't use them anymore. So the trick is to keep your calorie intake more steady. Try not to have to burn fat in the first place, and of course, don't get overweight, because then you've got too much fat around, and eventually you're going to want to lose that. For you long chain fatty acid oxidation folks, it's a little bit more complicated because we're giving you a fat that bypasses your block. But you need at least 10% of your calories from fat to keep from becoming depleted on those fats that you can't make, those fats called essential fatty acids. So while the fats that you eat aren't toxic per se, it's hard not to eat too many calories from fat if you're getting 15 or 20 or 25% of your calories from MCT oil, and now you take a regular diet that's got 35% of calories from long-chain fat, way too many calories from fat. So we cut your fat back, your long-chain fat back, to try to balance you a little bit more. Let's see. We have Nathaniel asking, does that mean MCT oil as a treatment isn't sustainable long term? Well, it's all we've had up till now, so it's been sustainable for this long, and it helps some. But I think I've shown you that it isn't, that there are some problems with it, and we think that our data are convincing that triheptanone is better than MCT oil for the reasons we've discussed. As we hope it becomes approved by the FDA in the coming year or so, I think it is certainly worth discussing with your care providers switching from MCT to triheptanone. How do you define the difference between muscle pain and rhabdo, and what is the CK threshold? Well, muscle pain is just that. If you have cramps, but the muscle inflammation or energy deficit isn't enough to actually cause your muscle to break down, then it's just cramps. It's muscle pain. Think about, well, I can tell you, if I tried to run 10 miles after this seminar, by tomorrow morning, my muscles would be achy. I probably wouldn't have too high a level of CK. That's what muscle cramps are, or muscle pain. To call it rhabdo, you actually have to have an increase in CK, the enzyme that is a marker of cell breakdown. There's an official definition, and some people say it's got to be above 1,000 to be an episode of rhabdo. Some say 2,000. Every lab has a different normal. Most labs are about 200 to 250. So if you're higher than that, by definition, you're having muscle breakdown. That's rhabdo. It may be subclinical. It may not be something that you notice. Many of patients with, especially the VLCHAD and LCHAD patients, you may have gone to your doctor, gotten a CK result back that was 1,000, and you said, "Gee, I didn't know it. I wasn't having any symptoms." So that's not symptomatic, but it's still muscle breakdown. So it's a matter of semantics. Let's see. We have Debbie asking, what's the riboflavin dose? Okay, I'm gonna take a pass on that one, Debbie, because I can't give you specific doses for specific diseases for specific patients. I will tell you that 100 milligrams of riboflavin is a lot. 300 milligrams is a ton, and if you take too much riboflavin, you end up with an upset stomach. So talk to your doctor and get a dose that works for you and your local care team. There is a new form of riboflavin out there that's microencapsulated, and so if you need to be on the higher end, it's a little bit easier on the stomach. So you might want to talk to your care providers about that. Brittany is asking, for patients who haven't had rhabdo, is the antioxidant therapy still going to be recommended? I guess the answer to that is really it depends, because remember, there are other symptoms in fatty acid oxidation disorders, the heart symptoms, the hypoglycemia. So the goal here is to bring everything under control. Since the triamcinolone has done a nice job of preventing hypoglycemia, we probably won't need to use anything other than that if all you have is hypoglycemia. Now, most patients with long-chain fatty acid oxidation disorders, as they get older, will have some muscle symptoms. And so in that case, if you have muscle symptoms and they're not being treated, whether you're on MCT oil or C7, one of these other drugs could be helpful to you. The cardiomyopathy, although it's improved with the C7s considerably, still isn't completely eliminated, and so that may be another set of symptoms that's going to ultimately be a very an indication for use of these, well, some of these other drugs. Our goal, when I talk about precision medicine in treating these disorders, is not to look at any one of you and assume that you're the same as anyone else. It's to be able to take the best information that we can from you, and I may be able to do that from blood. We might be able to do that from your blood cells. We might be able to do that from a little skin biopsy, very minor procedure done in the office, no stitches, just a little numbing medicine, and we can get a lot of information from that. Whatever we need to do to get our information about you and your mutation and why you have your disease will then allow us to look at ultimately our whole set of potential therapies and pick the one that works best for you. And there's always going to be a little bit of trial and error. We try one, if it doesn't work, we try another. Combinations that we know work best in cells, and ultimately we hope that we can translate that into what your flavor of disease is and fix you, as opposed to trying to make you look like everybody else that we treat. So I don't see any other questions. I'm going to just end by saying that this is really a very, very exciting, one more just popped up. I'll come right back to you, Deb. Very exciting time in fatty acid oxidation disorders. We're moving from not having any approved therapies to getting our first approved therapy, fingers crossed, and ultimately multiple therapies, and some combination we hope will ultimately not just make your symptoms better, but give you a perfectly normal life that is symptom free. Let's see. Deb asks, do all the long chain disorders show a significant benefit from C7, or are there some of them that don't benefit as much? I think it's fair to say that everybody that I've treated with it has felt like they've done better. And the problem with that is it's subjective. You know, we try to make that as scientific as we can. We give our patients quality of life surveys that they fill out for us. There are a limited number of patients who seem like they have no effect at all, and those are going to be the ones who are going to need something else. And there are also individuals who have had symptoms that have been going on for so long that they've got damage, in particular the heart. If you've had cardiomyopathy for a long time, you end up with what's called fibrosis, and your heart just can't recover, even if we can fix its energy. So we've had patients who have needed heart transplants. We've had patients who haven't survived because their hearts have been so damaged, even on C7. So nothing is 100%, I guess, is the way to answer that. And Brittany snuck one last question in there. Okay, we'll take that one and then sign off. Will skin biopsy be required? I hope not, but if it is, we're going to try to do some of these same studies on white blood cells. But I will at least tell you that this is if you ask me what's easier, drawing blood or doing a skin biopsy, it's actually the skin biopsy. Because I can see your skin, I can't see your blood vessels most of the time, and it's very easy to just go in. It's a little cookie cutter type of an instrument. Looks like the end of a pen, a ballpoint pen, with the point retracted. That's the size of it. So we just do a little bit of numbing, depending on your age. We sometimes do it here, sometimes we do it on your thigh. Some people do it on the backside, and you just put a little bit of numbing cream on there, a little bit of lidocaine. And use this tool for the biopsy. Put a Band-Aid on it. In a couple of days, it's better. So to get the most benefit from these new drugs, will want to know how you respond. But it's not going to be required as of yet. Oh, you keep sneaking those questions in, guys. Peru asks, we suspect mitochondrial disorder based on an acylcarnitine test. How do you rule out fatty acid oxidation disorders? Got to go see a metabolic doc for that one. They're trained to make those diagnoses. It's a combination of blood tests and DNA tests, and sometimes that skin test that I talk to. Oh, we got a few more minutes here. You're getting into it, guys. We got some more questions coming up here. Brittany, how do you feel about symptomatic carriers on patients who had carriers who are severe mutations? That's a really tough one. So there are lots of I've heard lots of reports and had families say to me, my child has LCHAD deficiency. I've always been a little slower, or I have cramps, or I have symptoms that I can't explain. We don't know. It's possible that you could have a mutation that affects your house of cards in a little bit, but doesn't knock it out completely, and may leave you with some mild symptoms. I think we have to put that one down as a to be determined, and as we get better at treating the patients who we know have problems, we'll also be able to look back on those parents and maybe try a drug and say, let's see if you get better. And if you get better, then we have to assume that the problem was related to that mutation in the first place. Let's see, Kimberly asks, will you please elaborate on the drug cardiolipin, where the positive effects found in the VLCAD mouse? We haven't gotten there yet. So the drug isn't cardiolipin, it affects cardiolipin. And just so you know, this is a I don't know, should I say dog eat dog business, or maybe a mouse eat mouse business? I'm going to go right back from here to my office to write the grant that lets me work on those cardiolipin experiments. The NIH funds these experiments, and that's how we get to do them. So we've only tried the cardiolipin drug on cells so far, but you're one step ahead of me, and I'll try to catch up with you maybe by the time we get to the next webinar. Let's see, what's the Can't see where one of them just rolled past me. Here it is. What's the most effective way to test for these mutations for your genetic disorder? And that's from Doba. Again, talk to your metabolic doc or your geneticist. It's a very easy test. It's a blood test. Send it off. There are lots of labs in the United States that can do this test. Very easy to get it done. They actually do check the DNA sequence. They read the code of the gene for the disorder that you have. Does one have to go to Pittsburgh to participate in clinical trials? That's Margarita. Some yes, some no. That MCAD trial is only going to be done here. Maybe we'll get some other partners for that as we get further along. Most of the other things that I'm talking about are multi-center trials. You can find that information by going to clinicaltrials.gov, search for fatty acid oxidation disorders, and it'll actually tell you what sites are doing it, give you the indications, what the entry criteria are, so you can get a lot of information there. We're also going to be mirroring that, or we'll be pointing you to that off of our informnetwork.org website. Do you think gene therapy or editing will be used for these conditions? Well, that comes back to my closing comments that once we can figure out how to get all of those all of the tissues that need to get treated at the same time, then the answer is probably yes, but not in the immediate future. 10 years is a lifetime in genetics, and so I can't tell you what's going to happen in 10 years from now, but it's certainly not going to be 5 years from now. Some of these other things that we've been talking about are likely to be available to you in the 3 to 5 year time frame, not gene therapy or gene editing. That's going to be a lot further down the road. So let's see, to confirm, for a patient with no previous cardiac issues, C7 can still improve normal cardiac function, and that's from Sharika, and the answer is, in our study, yes, it improved. So these were patients who had no evidence of cardiomyopathy, or were not said to have cardiomyopathy on their baseline echocardiogram by the person reading the echo, and their cardiac function got better on C7 compared to C8. Will the phenylbutyrate trial admit children? From Margot. I think we're approved down to for that trial down to 14, Margot, but I can't remember. I'm going to have to go to clinicaltrials.gov. We're trying to get it down as quickly as we can. It's a drug that we use routinely in babies for different disorders, so it's safe. But the FDA requires us to work our way down. I think we probably only have to do one more group of older patients before we move it down into kids. To sort of take your question one step further, it's the young kids that we worry about the most, the ones who can't tell us they're feeling sick or don't know enough to eat through an illness, and those are the ones we'd like to treat the most. And so the stakes are the highest there, and we will certainly try to move this as quickly as we can down. Let's see, Brittany, you're busy. Does that mean that someone can be in rhabdo without elevated CK? No, you can have muscle pain and not be in rhabdo. If you have elevated CK, that's the definition of rhabdo. So just a definition, but you can have muscle pain without elevated CKs. And Debbie, which disorders does C7 treat? That's all the long-chain fatty acid oxidation disorders, from CPT1, carnitine acylcarnitine translocase deficiency, CPT2, VLCAD, complete trifunctional protein deficiency, and LCHAD deficiency, all amenable to treatment with C7, and have been all of those disorders have been treated with C7.
Dr. Broccoli, it's almost 3 o'clock, but if you want, we have lots of questions.
We can. If you haven't heard that, my little fatty acid oxidation whisperer out here has said that it's already 3 o'clock and asked if I could stay for 15 minutes because we have more questions. Yeah, I'll hang out for 15 more minutes. So go ahead.
15 more minutes. Okay, we'll send that.
Excuse me, I'm going to take a sip while we're waiting. All right, they've got to transfer them from your questions over to something that I can see. In the meantime, just to give you some time frame about how things are going to likely play out here, we will have the MCAD study up and running hopefully by the summer. It's a good time to get college kids and you high schoolers who can take time away in the summertime to us. The Reneo study, the transcriptional activator, we think will be enrolling by April. That's going to be in 4 sites, and let me see if I can remember them: Oregon, Salt Lake City, Chicago, and here. So you'll be able to go to one of those 4 sites. The C7 trials are open only at this point for compassionate use. If you are having a lot of trouble, the language that we use is failing conventional therapy. We can still, or one of the C7 sites can still sneak you into that study, but you need to contact the site study director and talk to them about your problems and see whether or not they will meet the definition of failing conventional therapy. The FDA may ask for an additional study. I've been talking about approval. They may ask for an additional study. So if they ask for an additional study, then we're going to have to come back to you and ask you to enroll in that study. If it gets approved, they'll ask for follow-up studies. And so there may still be some opportunities for studies after the fact, but of course then it also will be commercially available. Okay, I got them running again here. Let's see what we have. Will triheptanone be considered a pharmaceutical drug or a nutritional supplement? It will be pharmaceutical, so it should be covered by your prescription plans once they've had a chance to review them and get them on formulary. So the second it's approved, you got to talk to your health plans and ask them about coverage for that as a medication. Let's see. Why not use small amounts of MCT oil and C7 instead of just one or the other? And that's Stephanie. Well, there are 2 issues there, Stephanie. Number one is that the piece of C7 that's novel that we're trying to get is the C3. We can get a lot of C2 from triheptanone, but we can only get C3 from triheptanone. So if we give you MCT oil, it limits the dose of the C7 that we can give you. And we think we're working pretty close to the optimum dose. If we drop it too far, it's not effective. The other answer is that MCHAD, the drug that, or the enzyme that metabolizes C8 and C7, works better with C8 than C7. So if you've got a lot of C8 floating around, it actually interferes to some extent with the C7 metabolism. So C8 plus C7 doesn't make sense for a couple of reasons, and actually is probably counterproductive. Let's see. What do you mean triheptanone can improve symptoms of cardiomyopathy? Does it stop the thickening of the walls? Is propranolol enough for hypertrophic cardiomyopathy? And that's Iris. Iris, we've had patients with cardiomyopathy that have started on C7 and their hearts go completely back to normal. So these are, you can have 2 kinds of cardiomyopathy with fatty acid oxidation defects. So your heart can sort of get weak and just end up being like a balloon filled with water. So it stretches, but the wall stays thin. That's called dilated cardiomyopathy. If the wall stays thick, that's hypertrophic cardiomyopathy. And both seem to be responsive to C7. Now, not everybody gets to 100%. Normal activity, but most of our patients that have had heart problems have gotten better. If you're better on C7, you don't need the propranolol. If you still have some heart dysfunction, then you may need another drug, and propranolol may not be the right drug. So this is one where you really need to go to somebody, a metabolic physician who's working with a cardiologist who knows these diseases and can give you the right medication. Let's see. Nicole asks, is there always a mutant gene responsible for FAODs? Our 10-year-old has had muscle weakness and pain and hypoglycemia with ketones during fasting and illness during both. And I'll shorten it up to say you don't know what he's got. And that's an all too common problem, unfortunately, Nicole. The fact that your son is making ketones would suggest it's not a fatty acid oxidation disorder, because fatty acid oxidation disorders, typically you don't make a lot of ketones, and so it could be something else. Not all mutations are discoverable by DNA testing. Typically, we find about 90% of them, and so what that means is about 2% of people have what can be diagnosed as fatty acid oxidation disorders for other reasons, using other tests, but don't find the mutation. But those other tests should be abnormal. So your metabolic doc should be able to look at your acylcarnitine profile, your urine organic acids, and do this skin biopsy test that I talked about, and tell you whether or not you have a fatty acid oxidation defect, whether or not there's a mutation that's identifiable on your on testing. You also asked about mitochondrial dysfunction, responding well to CoQ and creatine. So remember back in the beginning, I talked about how all three of these cycles interact. You can have secondary respiratory chain deficiencies in fatty acid oxidation disorders. You can also have secondary fatty acid oxidation disorders in patients who have respiratory chain deficiencies. Your constellation of symptoms and your response to therapy sounds a little bit more like a mitochondrial respiratory chain deficiency, so the other testing, if it's already been done and says it isn't a fatty acid oxidation disorder, is probably right. It's probably not a primary fatty acid oxidation disorder. I'll say this: if you send questions into informnetwork.org, I can't answer specific questions about specific symptoms or problems, but so I have to try to keep it general. Let's see. To confirm, for a patient with no previous cardiac issues, C7 still improves normal cardiac function. Can you review that snippet you mentioned? Oh, I think this one is showing up from a previous. That showed up before. I think that was one of Stephanie's questions. Is it possible that it could work to prevent future cardiac issues? I don't know if I answered that half of the question. The answer is we hope so, but we haven't done long enough follow-up to know. So we've got now patients who have been treated for almost 20 years, starting with original Dr. Rose studies. I've followed patients for now, well, I actually followed Dr. Rose's original patient, so I know that patient. And we certainly hope that our patients that we're identifying through newborn screening, if we start them on C7, will ultimately not develop cardiomyopathy. One of the things that the FDA requires when a drug is approved is something called post-marketing studies, and those studies ask those kinds of questions. So they'll say you got to watch these patients for 10 years and see if anything happens. It's part of studying a rare disease. Son is interested in trying the keto diet. Yes, you cannot use the keto diet with fatty acid oxidation disorders. Fats are what you can't burn. So if that's what you give, it's going to make you horribly sick and could kill you. Stay away from it. They don't usually be that blunt, but don't do it. New guidelines for standard LCHAD diets. That's Michael. And resveratrol is an antioxidant. We've tried resveratrol. It hasn't worked in cells. So there are a couple of investigators that have reported that it improves cells by a little bit. Resveratrol is one of those transcriptional activators that also has a little bit of direct antioxidant function. So that's one of those to be determined, whether or not it'll be as effective or not compared to some of the other things. We think the drugs that we're using are more potent on both sides of that equation, and so are going to be a better drug than resveratrol. New guidelines for VLCAD diet, yes, coming out soon. The GMDI, the Genetic Metabolic Dietitians group, is currently preparing, and I hope soon to publish, new guidelines for VLCAD diets. Those are going to work pretty well for any long-chain fatty acid oxidation disorder, but they're only going to be validated for VLCAD. So keep your eye out for those. Your metabolic physicians and dietitians will certainly know about them being approved and be able to bring them to your attention. More C7 and cardiac questions. This is Kimberly. Kimberly, the metric that we used was cardiac. The hearts were able to do more work at a lower heart rate. So that's kind of a general measure of overall function. And that was in the study that Dr. Gillingham and I did. In the triheptanoin trials with Ultragenyx, we're actually following echocardiograms, and so hearts that aren't beating well, usually measured by something called the ejection fraction, start beating better. So a normal ejection fraction is 65. We've had patients with as low as essentially 0, and they go back to normal, 65, when they start on triheptanoin. Do all patients with long-chain fatty acid oxidation disorders have cardiomyopathy? No. Right now, we don't have good natural history data. It's one of the things that we're trying to capture with the INFORM network. Probably 10 to 20% as a guesstimate. And that's because we have most of our history with kids or with patients who are less than 20 or 25, the era of newborn screening. For adults who are now starting to hit their third and fourth and even fifth decade of life, with especially VLCAD and LCHAD, we just don't know whether you're going to be at risk for heart failure later on. Job safety for me. I hope you'll continue to go see your metabolic docs. It's going to be important to follow that and keep tabs on it over time. Stephanie again, any benefit for patients to get their own cytokine profiles to know how much inflammation they have? It's still an experimental test, Stephanie. I mean, the test itself is well standardized, but we don't really know how to interpret it in all cases. So I'm using that test on my patients on a research basis, but I can't really recommend it as a routine clinical test, because your docs are just not going to know what to do with it. Once maybe some of these other drugs come down the road, there may be some benefit to doing that. Is it even possible in the average clinic? I would say there are no average metabolic clinics. So if you're in a metabolic clinic, you're in a tertiary care medical center, and they should have access to that technology. Do you recommend a child with MCAD have a liver checked every few years? That's from Lisa. That's another one of those that we don't have really good answers for. We know that adults with MCAD deficiency are less prone to have episodes of metabolic decompensation as they get older. We don't have any good evidence that they have a higher risk for liver disease as they get older. And so as children get to be adolescents and adolescents get to be young adults, it's certainly reasonable to space out the timing on visiting your physicians. I'm delighted if my patients with MCAD deficiency want to come and see me once a year, but if they're coming from a long distance and say, look, you don't do anything but examine me and tell me, okay, come back next year, I can see the argument for going every couple of years, but I don't think you want to let it go any longer than every couple, 3 years, if for no other reason that you're hearing that there's lots going on in the field, and so keeping abreast of the new technology and new studies, something that you can do when you meet with your metabolic team. I don't know. I've either answered them all or they're behind me on the typing. It is quarter after though, so folks, I hate to say it, but I got grants to write and meetings to attend. So I really appreciate your taking the time to be on this webinar. I hope you found it helpful. Please feel free to get involved with Inform Network through the informnetwork.org. There will be volunteer opportunities there for you to help move the field of fatty acid oxidation disorders along, we'll have opportunities for face-to-face meetings as we move that consortium forward. So thanks for participating, and I hope to see you on the next webinar. Bye-bye. We're done. Hey.